Om Swastiastu, Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Hello everyone, so we are from small group discussion B9. And now we want to present our student project assessment that the title is Method for Detection of DNA Mutation in Marfan Syndrome. And this is all our student member list. And let's start from the introduction.
My name is Widiantara with my student ID number of 200251207. I'm going to talk about the background information contained in the student project. As we know, humans have a lot of differences in status. It is caused by a double helix shape comprises of nitrogenous base, sugar sulfate backbone, and delcirribose that controls a lot of protein synthesized inside our body, called DNA. How can the body manage all of this protein synthesized perfectly? Actually, it isn't that perfect. When there's a mistake or reaction that changes the DNA, it is called as a mutation, and it could pass down to the next generation. In the student project, we featured Marfan syndrome. What causes this syndrome is the mutation of Phoebe gene, existed chromosome 15. This mutation had impact on the fibrinogen, which is one of the essential extracellular matrix components. Next.
Okay, hello. Before I continue the presentation, let me introduce myself first. My name is Evangelina Vincent Lacerda with student ID number 2002511170. So in this slide, we're going to talk about the problem formulation and the objective. The problem formulation is what does the DNA mutation means, what is Marfan syndrome and the current understanding of it in the community, what is the description of methods available to detect mutation of Marfan syndrome. And the objective is to provide an explanation to the reader about the DNA mutation in general, to give a better understanding about Marfan syndrome, to acknowledge reader and describe more detail about some of the method to detect mutation of Marfan syndrome. Next. So the benefit that we will learn is we will understood about the DNA mutation in general, Marfan syndrome, and some methods available to detect mutation of Marfan syndrome. Next.
My name is Phoebe Ezkiella and my student ID is 2002511169 and I'm going to explain about types of DNA mutation. First of all, mutation is an alteration in the genetic material of a cell that can be permanent and transferred to the cell's offspring. There are two types of DNA mutation, such as point mutation and frame shift mutation. Point mutation happens when there is a change in the single nitrogen base in the DNA sequence and is usually the least harmful type of DNA mutations. Point mutation can be classified broadly into two types, which is transition and transversion. If a nitrogen base is substituted with the same type, transition will occur. Meanwhile, when purine base are substituted with pyrimidine base and vice versa, this is called the transversion mutation. This can cause three conditions such as missense, nonsense, and silent mutation. The other type is frame shift mutation. It's a particular type of mutation that involves either insertion or deletion of extra bases of DNA. Insertion is a situation when a single nitrogen base is added and causing the frame to be pushed down by a latter, creating a completely different proteins. Deletion is an occurrence when a nitrogen base is taken out of the sequence, also causing the reading of the frame to shift. As a matter of course, a drastic change in amino acid may cause severe problems or even death if an essential protein is altered. Next.
Okay, my name is Herry with student ID number 20025111189. I will continue the presentation about Marfan syndrome. Marfan syndrome is an inherited disease that caused by dominant autosomal condition. The prevalence of this disease is 1 in 10,000 or 20,000 individuals. The disease is associated with a mutation in FBN1 gene, which encoding fibrillins, and sometimes with mutation at TGFBR1 or TGFBR2. The fibrillin is an important protein to the extracellular matrix, which contribute to the structure of microfibril, so that the mutation in FBN1 will affect some manifestation disorder associated to the skeletal system, cardiovascular, pulmonary system, ocular system, skin, integument, and dura ectasia. Next.
My name is Nathanael Putri, with my student ID number is 200254168. The manifestation of Marfan syndrome to the skeletal system is overgrowth, which can affect the anterior chest deformity or magnetic chili at rest time. Marfan syndrome manifestation is also associated to duroextasia or widening of dural root or sac, which is occur on 63 to 92% people with disorder. The pulmonary system, it can affect spontaneous pneumothorax and apical blast. In the cardiovascular, it can cause a prolapse of either the mitral valves or both anteroventricular valves. In children, it can cause pulmonary hypertension, congestive heart failure, and death in infancy as a result of insufficiency of the mitral valves. In the skin and integument, diseases can cause asthma without relation to weight gain, pregnancy, stress, and incisional hernia. The effect of Marfan syndrome in ocular system is ectopial lenses, abnormally flat cornea, increased axial length of globe, and decreased meiosis. Next.
Thank you. My name is Putra from Sari. My student ID number 20025111177. The next method is PCR. PCR or polymerase chain reaction is an DNA replication method that works enzymatic without using organisms. It was found in 1983 by Herry. This technique often used in many science fields such as biochemistry, molecular biology, and etc. In this method, there are three things that can be called as a key or main ingredients of a PCR reaction. Those are take polymerase, primers, template DNA, and nucleotides as a building block DNA. In this cycle include three steps, which are melting steps or denaturations, annealing, and extension. Will be continued by my friend.
My name is Ardisa Pradasuari. My student ID number is 200211172. So I'm gonna explain how does the PCR detect the Marfan syndrome. This method has been used in order to detecting many diseases and syndromes. One of those is made by following the reverse gen criteria to perform monoallelic sequencing. Exon 63 was amplified from DNA extracted from blood using polymerase chain reaction, or what we often hear with PCR. With oligonucleotide primers for 35 cycles with fusion high fidelity DNA. The PCR product was then purified and carried out into cycles of amplification with DNA polymerase tech before being cloned into the GAMT easy cloning factor and converting Escherichia coli.
So my name is Putu with student ID number 2002511202, and I want to explain about MLPA, a way to detect duplication and deletion of DNA. So MLPA is multiplex ligation dependent probe amplification. It is one way to detect these abnormalities. The detection is done by detect large deletion and duplication in FBN1 gen. MLPA can detect the duplication and deletion of DNA on Marfan syndrome. There's also advantage and disadvantage of using MLPA. The advantage of using MLPA is capable of re-engineering small molecule, and it also capable of handling up to 40 targets with high throw output and incur low cost. And the disadvantage of using MLPA is lack the ability to measure the missing neutrality of heterozygosity and can cause problems with donor heterogeneity, mosaicism, or contamination with normal cell.
My name is with student ID number 20025111193. So I will explain about there are 5 steps of the MLPA reaction. The first of all, the denaturation of the DNA and the prop hybridization. And then the second step is ligation reaction. The third step is amplification of the PCR. And then the fourth step is the process of separating the amplification product using electrophorese. And then the last step is perform data analysis. And then MLPA can detect duplication of FBN1 and the TGFBR2 using MLPA PO65 and PO66 kits. Next.
Hello, my name is Baruna with student ID 20025111770. So I'm going to explain about the ion current personal genome machine, PGM. So peripheral blood tests were gathered from 3 MFS patients and an ordinary control with educated assents. Genomic DNA were disengaged by standard strategy and afterward exposed to focus sequencing utilizing an ion amplisec TM inherited disease panel. 3 multiplex PCR responses were done to intensify
the coding exons of 228 qualities. DNA parts from various examples were ligated with barcode sequencing connectors. Layout planning and emulsion, PCR, and ion pair particles advancement were done utilizing an ion one-touch framework. The particle circle particles were sequenced on a 318 chips utilizing the PGM stage. Information from the PGM runs were handled utilizing an ion torrent 3.2 programming to produce arrangement for choose. After succession, arrangement, and extraction of SNPs and indels, all the variations were separated against DBSNP137. DNA groupings were envisioned with an integrated genomic viewer. Next.
Okay, my name is Bagus. My ID number is 2002511184. So, as you can see, graphic analysis, machine learning, classification, and grouping is a meaning of multidisciplinary computer science, or as known as microarray gene expression data processing. So, microarray can measure several hundred of thousands of gene expression in one trial of an experiment. Gene expression level can also determine disease progressions and correlated genes, which can aid in early diagnosis and the prognosis of various types of cancer. Thanks.
Okay, my name is Kirana Devisavita. My student ID number is 20025111197. Microarray sequencing is designed based on the reference sequence from the ensemble database and the candidate nucleotide substitutions discovered by the microarray-based reorder system. Next.
Hello, my name is Wulansiwi Wahyu Widiantara with my student ID number is 200251111173. So the conclusion of this student project is DNA regulates heredity through the formation of RNA by transcription process and the formation of amino acid in the translation process. Sometimes in the process of formation, some errors occur which result in damage so that the DNA is not protected and is called DNA mutation. One of the DNA mutation is Marfan syndrome. Marfan syndrome is caused by the mutation at FBN1 gene exists in chromosome 15 and affect the connective tissue. There are many ways or methods that can be used to detect Marfan syndrome, including PCR, MLPA, TGM, and a method that using high throughput microarray based on mutational analysis of fibrillin 1 gene. Thank you. So that's all that we can explain about our student project assessment. And this is all the reference that we use for making the student project. And this is too. And last but not least, we hope this student project presentation give a lot of information and more knowledge for everyone. And we close it with thank you.
Thank you.
Thank you.